So this part of the lecture, we have already covered the first part of chapter 9, which basically just walked us through the anatomy of our skeletal muscle cells. What are all those parts and pieces? This part of the lecture today, we will be going through muscle physiology. I told you on Monday, I do a lot, I call it chalk and talk when I'm in the classroom because we have a chalkboard. Obviously on here, I don't have a chalkboard, but I do have a whiteboard. So what I will be doing throughout this lecture is I will be sharing a whiteboard with you, and then I'll come back to the PowerPoint. Then we'll go back to the whiteboard. So there'll be a little bit of jumping back and forth between the two. So if you have some paper handy, you may want to grab a notebook, piece of paper. If you've got colored pencils, that's also pretty helpful. Lecture, I'm going to attempt to use different colors in the whiteboard if I can pull those tools up and make them work. Okay. So let me actually start with a whiteboard. All right, so you can see, we left off on Monday. I walked you through, let's see. I walked you through a video. Sorry, a drawing where I have the nerve coming into the muscle cell, and we have our muscle cell. So remember, muscle cells, we call them muscle fibers because they're really long, okay? We have T-tubules that dip down through that entire muscle cell. On either side of those T-tubules are something called terminal cisternae. And these store calcium. So I'll try to keep the terminal cisternae blue, and I'll also try to make sure when I write calcium, I write it blue in color. So these are storing our calcium. Now we also have within our muscle fibers, we have sarcomeres. And remember, a sarcomere runs from Z disc to Z disc. And remember, the Z discs are just the proteins that are binding those thin filaments made of actin together. And your book usually has the actin colored in blue. And then we also have our thick filaments made of myosin. Remember, myosin has those golf club looking heads. Okay. All right, so there is my sarcomere. So we're going to walk through muscle physiology, which is how does a muscle cell contract? How does it get shorter? And we're going to do it from, I mentioned this on Monday, we're going to do it from 3 aspects. So when an impulse comes down a neuron, oops, it has to jump from a neuron into the muscle cell. So that's going to be kind of the first step that we're going to walk through. How does that happen? How does an impulse from your brain down a neuron, how does that jump from a neuron into a muscle? And then we're going to talk about how that impulse will spread down the muscle and down the T-tubes. So the second thing we're going to talk about is how. How does that happen? How is it spreading? And then once it gets into the T-tubules, calcium gets released and the sarcomere gets shorter. And so that'll be the third thing that we talk about. So I drew this out for you on Monday. So as we go through muscle physiology, I'll keep coming back and drawing on this whiteboard. And we'll start with step 1, then we'll do step 2, and then we'll do step 3. Now, for each of these 3 steps that I have labeled here, there are lots of steps that I'm going to walk you through. So it's a lot as we go through it. Now, what we'll do at the very end of the lecture today is we're going to take steps 1, 2, and 3, and we're going to put them all together. So I have a slide at the very end of lecture today that has every single one of these steps all put together, and then we'll draw it out all together. Okay. So let me go back to the PowerPoint. And so you can see here that this is just walking you through what has to happen in order for a skeletal muscle cell, muscle fiber, to contract. 1, It has to be stimulated by a nerve ending. So that step number 1 that I have on that drawing is how the impulse from a neuron jumps from the neuron to the muscle. Okay, stimulated by a nerve ending. You will always see when we draw out muscle physiology, I will always draw it where you've got that nerve coming in and then you've got that muscle cell. Okay. Number 2, that impulse has to be able to propagate along the sarcolemma. So remember, we have our step number 1 where the impulse comes down the neuron and jumps into the muscle. That has to happen. And then our step number 2, it has to be able to spread down the sarcolemma. And then the last one is you have to have a rise in calcium levels inside that muscle fiber. That's going to trigger the sarcomere to get shorter and the muscle cell will get shorter. So that's our number 3. So we are truly walking through muscle physiology in those 3 steps. Okay. Now, anytime we link the electrical signal that's coming down the neuron, okay, up here, anytime we link that to a muscle contraction, we call that excitation contraction coupling. So you may see that in your book if you're reading along. Now, one thing that all of our skeletal muscles have in common is they're all under voluntary control. That means your brain tells them when to contract. So skeletal muscles have to be stimulated by a motor neuron. Now, I know we haven't done the nervous system yet, so you might go, well, what's a motor neuron? We actually have 2 kinds of neurons. We have sensory and motor. Sensory picks up sensation, and they're going to send signals toward the brain so you can interpret the sensation. Think of senses. Think of your senses, right? Touch, taste, sight, sound, smell. That's all information coming from outside or inside your body going towards your brain. Motor neurons are doing the opposite. They're usually going to cause some type of reaction, some type of movement. And motor neurons are sending signals away from the brain, right? Exiting the brain. So we say skeletal muscles are always stimulated by a motor neuron. So that's a signal coming away from the brain into that muscle. Your brain is telling the muscle what to do. Now, once that neuron gets into the muscle cell, it's going to branch profusely. Sorry, once it gets into the whole muscle, it's going to branch profusely so it can hit all the muscle cells. And each of those little branches, we call those the axonal branches. And those form something called the neuromuscular junction. So neuromuscular, put those 2 words together. It's the nerve and the muscle, the neuron and the muscle. So it's where those 2 come together is our neuromuscular junction. All right, so I'm going to pop back to a whiteboard and we'll kind of jump back and forth between the whiteboard and the PowerPoint. All right, so I'm going to clear this one. All right, so again, you know, when we draw this out, I'm actually going to draw the neuron as yellow because your textbook usually will have a neuron in a yellow color. Okay, so there's my neuron. Okay, and then I'm just going to keep the muscle cell black. I think it just makes it a little bit easier. Okay, so there's my muscle cell, right? So that area between that yellow neuron and the muscle cell, so notice how they're not touching. That is called the neuromuscular junction. It's just that area between the neuron and the muscle cell. Okay, and so some of the things that we're going to find in the neuromuscular junction, we'll talk about them and then we'll draw them out on that drawing we just did. Okay, so one thing that we'll find is at the end of the axon, notice when I draw the end of that axon, I kind of draw it like this. I make it look kind of fat at the end. We call that the axonal ending, where it gets kind of big. And inside we'll have something called synaptic vesicles. And synaptic vesicles are basically just little vesicles, little bubbles, and inside they contain a neurotransmitter. Now, I talked to you on Monday about neurotransmitters and how these are chemicals that can jump between neurons and muscles. They can help a muscle contract. And the neurotransmitter that we most often find in the axons that are stimulating skeletal muscles is something called acetylcholine. Now, I pronounce it acetylcholine. Some people pronounce it acetylcholine. I say acetylcholine. That's just how I learned it. Doesn't matter to me how you pronounce it. A lot of times when I write out acetylcholine, I will write out ACH, right? So you can see that over here. I'll write that out ACH for short, just so I don't have to write the whole thing. Now we have like identified over 50 different neurotransmitters that we find throughout the body. Acetylcholine is just the one that we find within our skeletal muscles coming from those axons. Another thing is something called the motor end plate. This is the part of the sarcolemma directly across from the neuron that contains receptors for acetylcholine. And also you'll notice when I draw out that neuron and muscle cell, they're never touching. There is always a space between the two. And that space is called the synaptic cleft. Okay, so let's jump back to our whiteboard, to our drawing. I'm actually going to make my neuron a little bit bigger so that we can draw inside of it. Okay. There we go. All right, so there's my neuron. Now I told you inside that axon, we're going to have synaptic vesicles that contain acetylcholine. I will just draw those in orange. So here's my synaptic vesicles. And inside the synaptic vesicles, I'm just going to put some little dots in there. But those are synaptic vesicles. We can go ahead and label these. These have that acetylcholine in them. Okay. Now, I also told you that directly opposite from that axonal ending on the sarcolemma, you have something called the motor end plate. So it's really like this portion right here. It's actually sort of, it's not smooth. It's sort of a wavy area, and that is our motor end plate. And the reason why this is a special area of the sarcolemma is because it has receptors for acetylcholine on it. So I'm going to do those in orange since I did acetylcholine in orange. So these are little receptors. I make them look like little Ys. Okay, so here's my little acetylcholine receptors. That is the only place on a muscle cell that can actually grab on to acetylcholine. Why are receptors important? If you don't have any receptors and acetylcholine gets released, that muscle cell would never know. So these receptors are very specific. They only pick up acetylcholine. And when they grab on, that's how the muscle knows that the neuron is sending a signal. Okay. So there are acetylcholine receptors on the motor end plate. So I'll write ACH receptors. All right, so let's go back to our PowerPoint and we'll add in this drawing from your book. So even though we're doing our own drawing, I do like to show you the images from the book so that if you're reading your textbook, then you can still relate it to what we're drawing out. So in this image, you are looking at one skeletal muscle cell, right? We can see that skeletal muscle cell right here. You can see that neuron coming in. And I told you that the neuron is going to branch profusely. And so you can see all those branches. And right here, we're looking at an axonal ending where it is coming in to that muscle fiber, that muscle cell. And so down at the bottom, this image is just a blow up of that region. Again, that is what we call our neuromuscular junction. And so a couple of things that we can see, again, we can see that axonal ending. Again, it's in yellow, so that's why I drew it in yellow. We can also see this sort of wavy motor end plate in there. So you can see, again, it's only directly across from the axonal ending. And it has receptors for acetylcholine. Now the receptors for acetylcholine are these little purple things that they have drawn right there. Now we can also see in the axonal ending, we can see our little vesicles that contain acetylcholine. Again, acetylcholine in this image though are the little green dots. Okay. So those are just the parts and pieces that we're looking at. So now what we're going to do is we're going to go through that step number 1. So I told you we're going to break physiology into an impulse comes down a neuron and it jumps from the neuron to the muscle. That's step 1. How's that happening? So this is our step 1. How are we jumping from a neuron to a muscle? So you have all of those listed right here. I am going to open up the whiteboard and I'm just going to draw you through what's happening. Okay. So let's pull that up. I'm going to erase some of the things that are on here. Now, if you are drawing along with me at home, you can just start a new drawing. You don't have to erase anything because you may want to keep all this information right here so you know what everything is. Actually, I am going to erase all of this because I'm going to draw it a lot bigger. Okay, so again, we're looking at our neuromuscular junction. So we've got our neuron, that big axonal ending. And I'm just going to draw on this side that motor end plate. Okay, so I'm just drawing that neuromuscular junction really big right there. And we've got our synaptic vesicles with acetylcholine. Okay, now that motor end plate, remember, has receptors for acetylcholine on it. So we'll draw those. Okay. All right, so let's walk through those steps on the PowerPoint. How is a nerve impulse going to jump from a neuron into a muscle? So the first thing that's going to happen is the impulse is going to come down the neuron. Okay, so the brain is telling the muscle, hey, we're ready to contract. So here comes that impulse down the neuron. The next thing that's going to happen is calcium channels are going to open. Now, we know that calcium plays a huge role in muscle physiology. We actually see it play a big role in 2 different places. It plays a role in neurophysiology, so we're going to see it up here in the neuron. It also plays a role in muscle physiology, so we're going to see it down in the muscle later. So we know that calcium channels open in this axonal ending, and when calcium channels open, calcium rushes into the axon. Anytime you change the amount of calcium inside a cell, something's going to happen. You are going to get a change, okay? So an impulse comes down a neuron, calcium channels open, and calcium rushes into that axonal ending. That is going to cause a change, and the change is going to be that those synaptic vesicles are actually going to drop. They're going to migrate towards the end of that axon. So they move towards the end of the axon. Okay, when they get to the end of the axon, they fuse and then they release their contents through exocytosis. So y'all should have learned exocytosis in general biology. I think of exocytosis as sort of like a bubble that when it rises to the surface, it pops. That's kind of what's happening here. So these little vesicles, these little bubbles that have acetylcholine in them, they kind of migrate down towards the end of the axon, they fuse with the end of the axon and they pop and they release their contents into that space. That space between the neuron and the muscle is called the synaptic cleft. Okay, so they release acetylcholine into that synaptic cleft. Now, y'all should also be familiar with diffusion. Diffusion means we're going to go from a high concentration to a low concentration. And if we look at acetylcholine in that synaptic cleft, where we have a lot of it right now is up by the axon. So it is going to diffuse down towards the motor end plate. Okay, so through diffusion, acetylcholine is going to diffuse across the synaptic cleft. Then acetylcholine is going to bind to these receptors on that motor end plate. Once those receptors pick up acetylcholine, they know that that neuron is sending a signal, and now that signal can move, I'll do it in black, down the sarcolemma and down the T-tubes. Okay, so this is how an impulse has jumped from a neuron into a muscle cell. Truly, it's through the release of acetylcholine. Acetylcholine is that neurotransmitter. Okay, so an impulse comes down a neuron. That's going to trigger calcium channels to open, and calcium rushes into that axon. That's going to trigger synaptic vesicles to drop to the bottom of the axon and pop. They release acetylcholine into that cleft. Acetylcholine diffuses across the cleft, binds to those receptors. Now the impulse is triggered in the muscle cell, and it's going to spread down the muscle cell and down the sarcolemma. Okay. So let's go back to our PowerPoint. So I'm going to walk you through these steps. These are the same steps we just did. Okay, so this tells you, okay, calcium enters into that axon when that impulse gets to the end. That's going to cause synaptic vesicles to migrate to the end of the axon. Acetylcholine is going to get released through exocytosis. Acetylcholine now diffuses across the cleft and binds to those receptors. And binding of acetylcholine to those receptors is going to cause an impulse or an action potential in the muscle cell. Okay, so we walked through steps 1 through 5. Okay. The very last step that's on here I haven't mentioned yet. This very last step would be, okay, I just walked you through how do you get an impulse from a neuron to a muscle, right? Well, after the muscle has contracted, it needs to relax again. And the only way we can do that is if we get rid of acetylcholine. Because if acetylcholine hangs out in the synaptic cleft, it's going to trigger an action potential in the muscle over and over and over again. And you will just keep getting muscle contractions. And so after you've gotten that muscle contraction, we've got to get rid of acetylcholine. It has to get destroyed. And your body does that with an enzyme called acetylcholine esterase. You know, it's an enzyme because it ends in ASE, right? So acetylcholine esterase. This is the enzyme that degrades acetylcholine. It actually breaks it apart, and it makes it so that it's no longer useful in the synaptic cleft. And it prevents the muscle from continuing to contract and respond to that stimulus. Okay. So we've done the drawing. I've walked you through the steps. Now let's do the same thing. We're just going to look at it from the image in your book. Okay. So today, I'm sure y'all have already caught this. We're going to beat a dead horse. We're going to do all these steps over and over and over again. But I do think it's helpful. All right. So again, remember, we're looking right here at the neuromuscular junction. So we're going to blow that up really big. So again, what we'll see, let's see, I'm going to change my pen color. We'll make it black. We're going to see that impulse come down the neuron. Next thing that's going to happen, calcium channels open, right? And calcium rushes into that axon. That always triggers a change. In this case, synaptic vesicles are going to migrate towards the end of that axon. They're going to fuse to the end of the axon and pop, right? We call that exocytosis. They're going to release acetylcholine, you can see here, into that cleft. Acetylcholine diffuses across the cleft, and it's going to bind to these receptors, these purple receptors, at the motor end plate. Now this picture actually is showing you those little green acetylcholines binding to that purple receptor, so you can see that happening. And that's going to trigger the impulse to now spread down that sarcolemma and eventually down the T-tubules. Okay, so this is the same thing we just drew out, but it's just in the textbook image. Okay. Oh, I didn't mean for all that to be on there. All right. And so now what we're going to do is, remember that very first drawing we did, I told you we're walking through muscle phys in three big steps, right? How does it jump from a neuron into the muscle? How does it spread down the muscle? And then how does the sarcomere get shorter? So we just did, how does it jump from a neuron to a muscle? We did big step number 1. So now we're going to do step number 2. How does it spread down the muscle, right? So if we go back to our whiteboard, right, we sort of ended this with this part right here. So it's spreading down the sarcolemma, right? So this is where we're going to pick up. How is that happening? How is this spreading down the sarcolemma? What does that even mean? Okay. So this is what we call an action potential. So when I say it's spreading down the sarcolemma, what is spreading? An action potential. An action potential is just energy that's spreading down the sarcolemma or down a neuron. So remember, we start this whole process with an impulse coming down a neuron. An impulse is the same thing as an action potential. So that means the exact same thing. Okay. So I'll use those terms interchangeably. So in order to walk through this, in order to walk through what is an action potential, what is an impulse, we have to talk about the membrane itself. So we have some membranes for cells in our body that are excitable. Neurons obviously are one, muscles are another. And in order for it to be excitable, to carry an action potential or an impulse, it has to have something called a resting membrane potential. So we're going to start with that. We're going to start with when a muscle cell is not even contracting, what does it look like when it's resting? What's happening at the membrane? So we're going to talk about that. And then we're going to go through the steps of getting an impulse and spreading an impulse. We call this depolarization. There's our action potential, repolarization, and something called the sodium-potassium pump, which plays a really big role in this, okay? But we're going to start with our resting membrane potential. All right, so if you just look at a muscle cell that's not contracting, it has this sort of resting potential. The resting potential is that the outside of the cell is positive, has a slight positive charge. The inside has a slight negative charge. And that causes the membrane to be at negative 70 millivolts. So we're actually looking at what is the potential inside. And so I'm going to draw this on the whiteboard. And I'm going to clear this drawing. So if you're drawing out with me, you're going to want a new drawing for this. I'm not going to draw out a neuromuscular junction in this case. I'm just drawing out a muscle cell so we can see what's happening along that muscle cell. Okay, so there's my muscle cell. It's super long. And I'm going to write a number 1 over here because this is just our resting membrane potential. Okay. And again, resting membrane potential is just where the muscle, what it looks like, what this membrane looks like when you're not getting a contraction. And I told you that the inside has a negative charge. The outside has a positive charge. Okay. Another thing that we'll see, and I'm going to do these in different colors, is that there is more sodium outside the cell, and there is more potassium inside the cell. And that plays a really big role in an action potential. Okay, so you do need to know that. So our resting membrane potential of a muscle cell is negative 70 millivolts. So what is that? Volts, you know that voltage is the way that we measure current, electrical current. So muscle cells and neurons, they carry an electrical current. It is not nearly the strength of what's running through the wires in your home. Those are like 120 volts, right? If you flip your light switch on, that's like 120 volts. In your body, we're looking at millivolts. So that would be 1/1000th of a volt. So it's a very, very small amount of current. And we say it's negative 70 millivolts because the inside of that cell has a negative charge and the outside has a positive. Now that charge difference only exists right across that sarcolemma. Everywhere else inside that muscle cell is neutral, and everywhere else outside the muscle cell is neutral. So we only see a charge difference right across the sarcolemma, negative on the inside, positive on the outside. Okay. More sodium ions outside, more potassium ions inside the cell. So that is what the muscle cell is going to look like when it's resting, when it is not sending a signal. Okay, so now let's flip back to our PowerPoint. So what happens when it sends a signal? How does that happen? So the first step in getting an action potential or sending a signal or an impulse is something called depolarization. This is what happens when the muscle identifies acetylcholine in the synaptic cleft. So when an impulse comes down a neuron and it releases acetylcholine and the muscle picks it up, this is what happens. That sarcolemma becomes depolarized. So acetylcholine is going to cause this. So acetylcholine is going to cause sodium channels to open. Okay, so I'm going to go to, well, I'll walk you through this and then I'll go to the whiteboard. If we think about where we find sodium, so remember I just did that drawing for you and I told you there's more sodium outside the cell. Well, if you open a sodium channel, again, think of diffusion. It's a passive process, no energy needed. Sodium is always going to travel from high to low. You have more sodium outside the cell. So if you open a little hole, sodium's naturally going to flow into the cell. So sodium's going to enter into the cell. I want you to think about the charge that sodium has. This is an ion and it has a positive charge. So you are bringing a positive charge into the cell and it's going to cause the inside of that membrane now to become more positive. Now initially this is only happening where we have acetylcholine receptors. So it's only going to happen at that motor end plate. And this is why sometimes we'll call this the end plate potential. But if the stimulus is strong enough, so if that impulse coming down a neuron is strong enough that you get a ton of sodium coming in, we'll hit something called threshold. And threshold, I'll give you a number for it, is negative 50 to negative 55 millivolts. And this will then lead to an action potential, which we'll talk about next. So let's go back to our whiteboard and add this onto our drawing here, okay? So I told you that anytime acetylcholine is present, it gets picked up by those receptors on the motor end plate and it leads to depolarization. Right, and depolarization means that sodium channels open. Just that's it, sodium channels open. So acetylcholine's present, sodium channels open. And again, I told you that if sodium channels are open, so if I just do this, if I just put a little opening right here, remember we have more sodium outside the cell. So it's going to move from high to low. So sodium is going to move into the cell. Sodium has positive charge. So that is going to make the inside now become more positive. And the outside becomes more negative. Notice what we just did. If you just look at this drawing and you look at charges, look what we've done to those charges. We've just flopped our charges. So the inside was negative, now it's positive. That is an action potential. That's what you're doing. This is why resting membrane potential is so important. You flop your charges, that's how you send signals down muscles and down neurons, okay? So we say that once the inside becomes positive enough, we call that threshold. So if the stimulus is really strong, you have a lot of acetylcholine in the cleft, a lot of sodium comes in, you can hit threshold. And threshold is negative 50 to negative 55 millivolts. So I know that seems weird because I just told you that the inside, I drew positive charges inside, and now I'm telling you it's negative 50 to 55. I want you to think on a number line, okay? Do your number line up and down and put negative 70 in there and then put in negative 50. Negative 50 is more positive, right, than -70. It's closer to 0. And so this is why we say the inside is getting more positive, okay? So if we can hit threshold, if you have enough acetylcholine, if that impulse coming down the nerve was strong enough to hit threshold, then we can get an action potential. And that is our next step that we're going to walk through. Okay, so you have to hit threshold in order to move to an action potential. If you don't hit threshold, you won't get an action potential. So it's got to be strong enough to get you to that -50 to 55. Okay. Let's go back to here. So that next step is an action potential. And an action potential is if you hit threshold, now you're going to open up a ton of sodium channels. So a ton of channels open up. Sodium floods into that muscle cell and the inside gets really positive. That's an action potential. The inside gets up to positive 30 millivolts. So now we've gone from -70 all the way up to positive 30. That's a lot of sodium. So that's our action potential. And an action potential is self-generating. So what that means is if at the motor end plate, you hit threshold, you have enough sodium that comes in to hit that -50, 55, tons of sodium channels open, sodium rushes in, the inside gets up to positive 30, and now it's self-generating and it'll spread down the sarcolemma and down the T-tubes. Once you initiate an action potential, it can't be stopped. You will get a muscle contraction. And so if you have an action potential, you get a contraction. All right, so let's go back to our whiteboard and we'll draw our action potential in. Okay, so I told you that an action potential is if you hit threshold, okay, get enough sodium coming in, then an action potential will get you up to positive 30 millivolts. So the inside gets, I'm just going to do it like this, super positive. And the outside's going to obviously get more negative. And what's causing that to happen is all of that sodium coming in because you've now opened up a lot of sodium channels. Okay, and again, this is going to spread down the sarcolemma, down the T-tubes, you're going to get a contraction. So if we think back to that drawing that we started at the beginning of this lecture, when we have our neuron coming in and we have that synaptic cleft and we have our motor end plate, again, I told you that an impulse jumps from a nerve into a muscle through the release of acetylcholine, and then it's going to spread down the muscle cell, down the sarcolemma, and down the T-tubes. So these 3 steps just describe how that happens. Once acetylcholine gets picked up, okay, that's happening here. Once acetylcholine is picked up, we depolarize the membrane, we open our sodium channels. If there's enough acetylcholine to hit threshold, if enough sodium comes in, we get to -50, 55, we get an action potential. It's going to spread down the sarcolemma, down the T-tubes. Okay. The next couple of steps I'm going to walk you through, and I'll do this for every one of those big 3 steps that I talked about. The next steps I'm going to walk you through is, okay, we know that an action potential will eventually lead, you will get a contraction, right? Well, after you get a contraction, we need the muscle to stop contracting. We're going to need it to relax back out. So the next steps 4 and 5 are just how do we get that muscle to relax back out again? So after we've gotten our contraction, what are we going to do? Because we can't keep our membrane at positive 30. That means that you're going to keep sending an impulse and you're going to keep getting a contraction. Okay. So this very next step, I'll walk you back through the PowerPoint and then we'll do it on our whiteboard. So our next step is called repolarization. So after the action potential passes, now we have to repolarize the membrane, meaning we just got to get it back to -70. We got to get it back to resting. So I'll write that up here. It's just return. We need to return it to, I have no idea what's happening, to resting membrane potential. Okay, so we just got to get it back to that negative 70. Put that up here. So how we're going to do that? Now, at the very beginning, I showed you resting membrane potential, and I showed you sodium and I showed you potassium. More sodium out, more potassium in. How we went through depolarization and generating an action potential had everything to do with sodium. How we're going to get back to our resting membrane potential, repolarization, this has everything to do with potassium. So what we're going to do here during repolarization is we don't need sodium coming in anymore. We don't want the inside positive. So we're going to close our sodium channels. Those need to get shut. No more sodium coming in. Now what we're going to do is open potassium channels. And remember, we have more potassium inside the cell. So just like before, if we open some channels, naturally potassium is going to diffuse out. It's going to go from high to low, okay? Potassium diffuses out of the cell. Again, I want you to think about the charge here on potassium. It has a positive charge. So if we pump a positively charged ion out of the cell, if it's leaving the cell, now the outside is going to get more positive, and the inside is going to become more negative. And this will get us back to negative 70 millivolts. Now we say with this process of repolarization, it's going to happen in the same direction as depolarization. So basically, as an impulse spreads or as an action potential spreads down the sarcolemma and down the T-tubes, coming right behind it, we're going to repolarize. We're going to get it back to resting membrane potential. Okay. We have to get back to resting membrane potential before the muscle can be stimulated again, before you can get another contraction. Okay. And we call that the refractory period. So let's go to our whiteboard and we'll add this in. Okay. All right, so we have our step 4 here. Again, this is repolarization. I'm running out of room on my board. And during repolarization, like I told you, we don't want any more sodium coming in. Sodium has a positive charge. We already used that. We made the inside positive. What we're trying to do is get back to resting membrane potential. We're trying to get back to negative on the inside, positive on the outside. So what we're going to do is open potassium channels. Okay, and remember, if you look back at step number 1, we have more potassium inside the cell. So potassium is naturally going to diffuse out of the cell. Again, I want you to think about the charge that potassium has. It has a positive charge. So if we move a positively charged ion out, then the outside becomes more positive. The inside becomes more negative. Whoops, I didn't mean to do that. Inside becomes more negative. Get rid of that extra positive too. And this will get us back to negative 70 millivolts. Ta-da! We are back to negative 70 millivolts, negative on the inside, positive on the outside. So the whole point of repolarization is after you've gone through an action potential, you've gotten a contraction, let's get our muscle cell back to resting membrane potential. And it's really simple. We close those sodium channels and we open potassium. Now I did not in this drawing talk about closing those sodium channels, so let me add that in the drawing, right? So here's our sodium. Let's do this. Big X right here. We're going to close those channels. No more coming in. Okay. So this is essentially these 3 steps that are listed right here. This is what that muscle cell looks like before a contraction, what it looks like, step number 2, depolarization, as soon as it picks up acetylcholine. Ooh, now we're going to open some sodium channels. We're going to start to depolarize the membrane. If we can hit threshold, we get an action potential that's going to spread down the sarcolemma and down the T-tubes, and we're going to get a contraction, okay? And we've done that by just muscle cells and neurons can send signals. Just flop those charges. Once you get a contraction, we have to repolarize the membrane. We've got to get back to -70 so that we can go through this all over again. Now there is one more step that I didn't walk you through. And this very last step, oops, I need this to be black. This very last step is, I'm running out of room on my board, so I'll do it right here. The sodium potassium pump. This is on the PowerPoint, but I'll talk about it here and then we'll go to the PowerPoint. I want you to look at what has happened throughout this process with sodium and potassium. Okay, so just kind of look at that while I talk. The sodium potassium pump. This is something that you should have learned in general biology. Remember, this is a pump. It uses energy. It uses ATP, and it pumps 3 sodiums. Let me do it in our sodium color. It pumps 3 sodiums out of the cell, and it pumps 2 potassiums in to the cell, right? That's its job, right? So this is active transport. It's using ATP, pumps 3 sodiums out, so 2 sodiums in. Why is this important? Well, in steps 1, 2, 3, and 4, what we've done is we have altered our membrane potential, right? We started at -70. To get an action potential, we have to bring it up to +30. And then during repolarization, we bring it back to -70. So from an electrical standpoint, we're good. We're back at the right millivolts there. But from an ion standpoint, we're not good. I want you to look at where we start, especially we're going to start with sodium. I want you to look at resting membrane potential. Remember, we start with more sodium outside the cell. Where does sodium move during depolarization? Into the cell. Where does sodium move during an action potential? Into the cell. During repolarization, step number 4, sodium's still in the cell, right? So what we need to do is we need to fix our ion concentrations, and we need to get the sodium out. So this last step, the sodium potassium pump, look what it's doing. It's pumping sodiums out of the cell. It's fixing our ions. Let's do the same thing with potassium, right? If we start in step number 1 and look at potassium, we start with more potassium in the cell. During depolarization and action potential, is potassium moving? No, we're not losing any potassium. It's still in the cell, so we're good with potassium. But look during step 4, repolarization. Potassium goes out of the cell. We need to get it back in. So the sodium potassium pump is going to pump it back in. So that last step just fixes our ions. We're good in terms of our membrane potential. We're back at -70. We got there during repolarization. We just need to get more sodium out, more potassium in. And that is what the sodium potassium pump does. It fixes our ions. All right, so that's that last step, step number 5, sodium potassium pump. It just restores our ion concentrations, gets us back to more sodium out, more potassium in. So this image from your textbook is doing the same thing that I just did, that I just drew out for you. So let's do it here, but actually let's add our big step number 1 and our big step number 2 together. Okay, so remember, we've got an impulse that comes down a neuron. Your brain's going to tell your muscle to contract. When it gets to the end of the axon, calcium channels open and calcium comes in. That's going to trigger these synaptic vesicles to migrate to the end of the axon. Those little vesicles are like bubbles. They pop, release that acetylcholine. It's going to diffuse across the cleft and bind to those receptors at that motor end plate. Now remember, resting membrane potential is negative on the inside, positive on the outside. So this is what it's looking like until that acetylcholine is present. Once acetylcholine binds, it's going to open up our sodium channels, right? We can see sodium here, and sodium's going to come in. If enough sodium comes in to hit threshold, remember threshold is -50 to -55. Then a lot of sodium channels open, and the inside becomes really positive. Okay, we can see this wave of depolarization. The inside gets really positive. It gets +30. That's our action potential. And that's going to spread down the sarcolemma. You can see that positive on the inside and down the T-tubes. Now you can even see here, we can see our sodium channels are open, sodium's coming in. Look, potassium channels during this time, they're not open, right? Because we're generating an action potential. We want the inside to be positive. Now this little inset down at the bottom, this is trying to show you, okay, so after we've generated an action potential and it's spread, we got to get back to -70. And we're going to do that by closing our sodium channels. This is repolarization. And open our potassium channels. We're going to get the inside back to -70. So the inside's negative, outside's positive. Right? So this is the same thing. I think this one's a little more complicated to look at than doing the steps right together. But this one is kind of nice because it shows you the progression of the action potential down the muscle cell. Okay. This is just showing you resting membrane potential. It's just showing you what's happening with the voltage on the membrane. So you can see here that, let me move my controls, they're in my way. We can see that we've got over here on the y-axis, we have our, we have our membrane potential on the y-axis, and we have time on the x-axis. Now notice time is in milliseconds. This happens really fast. So it spreads not on the order of seconds, but milliseconds. So this is why you can get very quick contractions. It's not like it's taking a really long time to depolarize and get an action potential and repolarize and go through the sodium potassium pump. It's super fast. And so that's what this image is trying to show you. So you can see here that, again, we start at resting membrane potential. So we start at -70, okay? So we're chugging along at -70. An impulse comes down a neuron. Acetylcholine gets released, picked up at the motor end plate by those receptors, and that is going to cause those sodium channels to open. This triggers depolarization, right? And when sodium's coming in, the inside of that membrane's now coming up. It's becoming more positive. If it's positive enough to hit threshold, there's a number for threshold. Tons of sodium channels open, and you can see that membrane potential goes super high, up to +30. That's our action potential. That's our number for an action potential. You're going to get a contraction if you have an action potential. And so following an action potential, we have to repolarize the membrane. So we've got to close those sodium channels, open potassium channels, and the membrane will start to fall. That potential falls. And again, we call that repolarization. Okay, so we bring it back down to resting membrane potential. So this is just showing you how fast this process happens. So it's the steps I walked you through, but it's just looking at it a little different. Okay. So that's our kind of our step 1 and our step 2. So I told you that we were going to talk about muscle physiology from how does it jump from a neuron to a muscle? So we did that. How does it spread down the muscle and down the T-tubes? We just did that. So our third part is how does a sarcomere get shorter? Once an impulse spreads down the T-tubes, how is that causing the sarcomeres to get shorter and the muscle cell to shorten up? So this is our step number 3. Okay, and we call this excitation contraction coupling. So I'm going to walk you through that here and then we'll draw it out. Okay, so once that action potential spreads down the T-tubes, it's actually going to cause calcium to get released from the terminal cisternae. Now, this is the second time we've seen calcium play a role here. We saw it up in the very first step when that impulse gets to the end of the axon and calcium channels open, and here it is again. When that impulse spreads down the T-tubes, calcium channels are going to open from the terminal cisternae. Now remember, those terminal cisternae are little bulges next to the T-tubes, and they store calcium. Calcium is the key to unlock that chastity belt on that thin filament. So remember we talked about on Monday how our thin filaments are made of actin, looks like little beads. And actin has these little divots, these active sites. And that's what myosin wants to latch onto. Well, if you're not getting a contraction, you don't want myosin latching onto actin. And so there are regulatory proteins that sit on top of those active sites called troponin and tropomyosin. And I think of these as like the chastity belt for our thin filaments. So what calcium is doing is it is unlocking that chastity belt. It actually causes troponin and tropomyosin to move away from the active site. It moves off the active site. That's its job. Well, now the active site's open, and now the myosin head can latch on. So we call the myosin head latching onto actin, we call that cross bridge formation. And that is going to cause that sarcomere to get shorter. So this is what's going to lead to that contraction. The sarcomere gets shorter. So let me walk you through this on our whiteboard. Okay. And so because I lost you all, I have to redraw everything. I'm super sorry. All right, so we've got our neuron. We've got our muscle cell. Okay. Got my T-tubules. I like to do my terminal cisternae in blue because anything that has calcium in it, I'd like to draw in blue. Okay, now I'm going to do my thin filaments, right? My Z disc to Z disc is a sarcomere. Remember, we have lots and lots of sarcomeres within our muscle cell. Okay, there's my thick filaments. Okay, so I'm basically going to add steps 1 and 2 together, and then we'll walk through all those steps in that big step number 3. Okay, so remember we start with an impulse coming down a neuron. That's going to cause our calcium channels to open and calcium rushes in. That's going to cause all these synaptic vesicles that contain acetylcholine to migrate towards the end of the axon, and they're going to release acetylcholine out into the synaptic cleft. Now remember, on the motor end plate, we have acetylcholine receptors. And so acetylcholine is going to diffuse across the cleft, and it's going to bind to these receptors on the motor end plate. So remember, our resting membrane potential is -70. But as soon as acetylcholine binds to those receptors, we're going to start to depolarize our membrane. And so how we do that, remember, is sodium channels. So sodium channels open. That's depolarization. The inside of that membrane becomes more positive, and the outside becomes more negative. So we are depolarizing our membrane. If it becomes positive enough to hit threshold, remember our number for threshold is -50 to 55, we get an action potential. And that is going to spread down the sarcolemma and down those T-tubules. So there's that positive charge, that action potential, that +30, down the sarcolemma and down the T-tubules. Now this is where that big step number 3 comes in. Once that action potential gets to the T-tubules, it's going to cause calcium to be released from those terminal cisternae. When calcium's released, it's going to cause that chastity belt on actin, troponin and tropomyosin, to get unlocked. It's going to get moved out of the way. The myosin head is going to latch onto actin, and it's going to make the sarcomere squish towards the middle, right? So remember, in that middle, we have that A band, that dark band in the middle. And so we're going to basically lose that. So the sarcomere gets much shorter. Those thin filaments all move to the middle. and the sarcomere gets shorter. And if all the sarcomeres that are inside that muscle cell get shorter like this, then you get a contraction in the muscle, okay? So we have just walked all the way down to getting that contraction, right? And then we have these last two steps, which is just like with all of these that we've been going through, is undoing everything. So after you've gotten a contraction, again, you have to undo the contraction. And so the first thing we would have to do is remove calcium. If calcium unlocks the chastity belt, the last thing we want is calcium hanging around after we've gotten a contraction. So the terminal cisternae act like vacuum cleaners and they suck it back up. And that's going to cause troponin and tropomyosin to cover back over the active site. And myosin can no longer attach to actin, so the sarcomere lengthens back out and the contraction ends. Okay. So let's walk through this using the images from your textbook. Okay. So again, if we try to add all this in here, we've got an impulse coming down a neuron. Calcium channels are going to open. That's going to cause synaptic vesicles to release acetylcholine here into this cleft. That's going to get picked up by those receptors on the motor end plate. That's going to cause sodium channels to open. We call that depolarization. That's going to make the inside more positive. Initially, we call that depolarization. That happens only at the motor end plate. If it depolarizes enough to hit threshold, which is -50 to 55, we get an action potential, which is going to spread. And they show you this purple line right here. It's going to spread down the sarcolemma and down the T-tubes. Now, once it hits the T-tubules, and this image right here is just a T-tubule with the terminal cisternae blown up really big, so you can see it. So once that action potential, you can see right here, hits the T-tubules, that causes the terminal cisternae to release calcium. That's all these little red dots in here. That's calcium. So now you can see the calcium is coming out and it's flooding that muscle cell. So now we're going to look at up close what it's doing. If we can look at these little red calcium dots, again, remember these are the key to unlocking that chastity belt. So if we look at our thin and thick filaments at the top up here, you can see that chastity belt is still locked, right? You can see troponin and tropomyosin are covered over the active sites. But down here, you can see that they've been removed. And you can also see right here where calcium has bound to troponin. It has unlocked that chastity belt. It's moved it out of the way. So you can see those little red calcium dots unlocking those regulatory proteins, getting them out of the way. Now the myosin head can latch on. We call that a cross bridge, and it can push actin forward, and it can make that sarcomere get shorter. So there's a couple of terms that you need to be familiar with when it comes to actin and myosin and the myosin heads latching on. So one of them I've used, and that's this term cross bridge. So anytime myosin attaches to actin, we call that a cross bridge. Anytime myosin pushes actin towards the middle to get the sarcomere to shorten up, we call that a power stroke. And then anytime myosin lets go, we call that the cross bridge detachment. And then cocking of the myosin head means that myosin's sort of lifting up and it's getting ready to attach to actin. That's what that term means. And so I'm walking you through these terms because I want you to go back. Let's go back to our whiteboard just so I can show you. Right, so we've gone all the way through basically getting a muscle contraction, getting this sarcomere to get shorter. But nowhere in this drawing have I mentioned ATP, right? Other than like the sodium potassium pump. I haven't really mentioned ATP. I haven't written it down anywhere on here, but we know that muscle contractions use a lot of ATP. Where that ATP is used is in shortening the sarcomere. we are going to burn through a ton of ATP in getting a muscle contraction. So that's what I want to walk you through. I can't draw this out. Like, I'm not very good at drawing out all these the actin and the myosin, the thick and the thin filaments. And so I'm going to use some images, and we're going to talk a little bit about the ATP. Okay. So let's go back to our PowerPoint. And so that's what this image is showing you. It's showing you how ATP is being used in this process. Again, I just drew on our drawing ATP next to that sarcomere. So you should know that where we're really burning that ATP and using our ATP is getting the sarcomere to get shorter. So how are we using ATP in this process? And so your book has this great image. It is a circular image. So you can see that you just keep going around and around and around. In terms of when myosin latches onto actin and performs a power stroke, it pushes it towards the middle to get that sarcomere shorter. Each myosin head latches on, pushes, detaches, and then does it again. So it's like a ratchet. They sort of push, let go, push, let go. They just keep going through this process until the whole sarcomere is shorter. So it's not like a myosin head forms a cross bridge, pushes and lets go, and it's done. It keeps going. So this image is really good because it shows you that this is a cyclical process. Okay. It's also showing you where ATP is used. So ATP is essentially, I like to think of it as our energy currency. It's what we use for energy. And so ATP is sort of like cash, right? I mean, cash is great, but it doesn't do anything unless you're spending it. So it's kind of the same with ATP. I mean, ATP is great, but in order to use it, we have to spend it, right? So how we spend ATP, how we use it, so we actually break it down. The energy is in the bonds. So ATP stands for adenosine triphosphate. Tri means 3. So that means that there are 3 phosphates in ATP. And how we spend ATP is we break a phosphate off. And what we're left with when we do that is ADP, adenosine diphosphate. So I know this image is starting with a number 1 at the top. I actually start down here at the bottom. I think this one just makes more sense to me. So I want you to look, we have ATP right here, okay? And you can kind of see that myosin head is falling off of actin. So we are detaching a cross bridge. We add ATP, we detach a cross bridge. To spend ATP, again, I told you have to break a phosphate. And so when you spend ATP, it causes this myosin head to cock. It kind of gets ready. And how you spend it is you break ATP down into ADP and P. That's giving the myosin head energy to cock and form a cross bridge. So you can see it's latching on right here. All we've done to do that is we've just taken ATP and broken the bonds. We've turned it into ADP and P, right? It'll cock and form a cross bridge. To get it to perform a power stroke, get rid of ADP and P. Get them off of that myosin head. And now the myosin head will push that actin forward. That performs a power stroke. To get it to detach, add ATP. It'll detach. To give it energy, break ATP down into ADP and P. It'll cock and form a cross bridge. To do a power stroke, get rid of everything. It'll do a power stroke. So this is a cycle, okay? I'm going to star this slide because you have to know what's happening with ATP here. So I may ask you a question on the exam, and it might be something like, what has to happen for a cross bridge to form? And you would tell me, I'd have multiple choice, and you would tell me ATP has to be broken down into ADP and P. Or I might ask you, what has to happen to perform a power stroke? And you would tell me ADP and P have to be removed from the myosin head. Or I might ask you, what has to happen to detach the cross bridge? You got to add ATP. This is a little weird. It's kind of a weird way to think about it. But to detach the cross bridge, right, to add ATP, it does seem kind of weird. So the way that I remember this is I think of a condition called rigor mortis. Rigor mortis happens after death. It usually peaks several hours after death, and it causes the muscles to become stiff. You cannot stretch a muscle, you cannot contract a muscle, and that means that you can't move the patient, right? They can't be moved. You can't move that corpse. Like you can't bend the elbow, you can't bend the legs. They're stuck in that position. That's rigor mortis. And what's happening is that when someone dies, calcium floods their muscle cells. Again, I want you to think about calcium, right? Here's calcium right here. Calcium is the key to the chastity belt. So when you die, calcium floods your muscles. It causes troponin and tropomyosin to move off the active site, and you can form a cross bridge. But you can't detach a cross bridge because what you're not making when you're dead is ATP. So that cross bridge can't detach. And so your sarcomeres end up, you form this cross bridge and your sarcomeres get stuck in this position. Okay, you cannot detach that cross bridge. So this is how your muscles get stuck in a position. Now, eventually, you know, within 18 hours or so after death, your proteins, your actin and your myosin will start to degrade. And as that happens, then you can bend that corpse. You can move the elbows. You can bend at the joints again. Okay. Now it's not because they've suddenly started making ATP. It's because actin and myosin have truly started to degrade. Okay. So that's just, I mean, it's weird, but it is one way that I remember that to detach a cross bridge, you got to have ATP to do that. Okay. So one of the things that your textbook talks about is this sliding filament theory. And we've actually talked about it. I just didn't call it this. And so remember your sarcomere in a relaxed state, you have I bands and you have A bands. But when myosin forms a cross bridge and your Z discs get closer together and the sarcomere gets shorter, those filaments slide over each other. And that's what the sliding filament model of contraction is. It just talks about how the thin filaments slide past your thick filaments so that your sarcomere can get shorter. So if you're reading in your textbook and you come across this, that's all that means. Now, I showed you a video on Monday, not a video, I showed you during Monday, during the lecture, I showed you how I have a sarcomere model and I showed you that sliding happening. Again, I mentioned each myosin head is going to attach and detach multiple times during a contraction, like a ratchet. And so that's why this picture is a cyclical picture. Okay, it's not really showing a stop to any of that. It'll continue until the sarcomere gets as short as possible. And that's going to cause the muscle to get shorter. Now, this is also a nice picture because it's one I've already shown you, but this is showing you again the sarcomere. Here's the sarcomere again from Z disc to Z disc. It's a sarcomere that is in a relaxed muscle. So you can see our A band, which is here, right? A for dark. We can see our I bands over here, I for light. And you can see that there is room for those filaments to slide over each other. And truly all we're going to do is push those Z discs toward the midline. Okay, and you can see how the A band essentially, this A area right here just gets a lot bigger. We can see that we've lost this H zone in the middle is completely gone, and our I bands have disappeared on the sides. We no longer have those I bands. So the term contraction just means that we've activated cross bridges, and a contraction ends when the cross bridges become inactive, okay, and the muscle can relax back out. So I told you that when we started this lecture, I would take all those three steps, those three big steps. How does it jump from a neuron to a muscle? How does it spread down a muscle? And how does the sarcomere get shorter? And I would give you one slide that has them all. So every single step right here. So I like to tell students, and we're going to do this all together. I like to tell students that if you can walk yourself through every one of these steps in the drawing, you can answer every physiology question I ask. Okay, so if you can walk yourself through the steps, you're good in terms of what we just talked about today in this lecture. The only thing extra that you need, and I'll write this on here, the only extra that is not in these steps is ATP. So that is this slide. You have to know what's happening with ATP, and that is not in those steps. So that's the only thing extra. So if all you do is study this slide and you understand what's happening there, and you make sure you know every one of these steps, you could draw it out and you understand what's happening, you can answer all the physiology questions from chapter 9. Okay, so let's take all of these, and I tried to color code them like in purple. These are our big step number 1. And in blue, this is our step number 2. And in green, this is our step number 3. Now you'll notice at the end, it's kind of a hodgepodge down there. And that's because these are the steps at the very end of each of these where we're undoing what we did. And I put them at the very end. So what we're going to do is steps 1 through 9, we're going to walk all the way down to getting a contraction. And then steps 10, 11, and 12, we're going to walk our way back up and we're going to undo what we did. Okay, so let's pop over. This will be the last thing we do. Let's pop over to our whiteboard. So I'm going to clear this. We're going to draw it all back out from the beginning and just make sure that y'all can do it with me. Okay, so we've got our neuron. That should look super familiar. Okay, we're drawing out that neuromuscular junction. So we've got our muscle cell. Here's our T-tubules that go all the way through that muscle cell. We've got our expanded terminal cisternae. Remember, these store calcium. We've got our sarcolemma, Z disc to Z disc, right? Our thin filaments made of actin, and our thick filaments made of myosin. I'm also going to add in here our synaptic vesicles that contain acetylcholine. Okay. So you should be able to see, if you have a PowerPoint open, or some of you may have printed off your notes, you should be able to see our steps. So we are truly just going to walk through these in order. Okay. So this is our 1 through 9. So an impulse is going to come down a neuron. The next thing that's going to happen is our calcium channels are going to open. Calcium's going to rush in. Told you that anytime you change the amount of calcium inside of a cell, something's going to happen. In this case, synaptic vesicles migrate towards the end of that axon. They fuse and they release their contents through exocytosis. Remember, it's like a bubble popping. Now remember, we do have acetylcholine receptors on that motor end plate. So acetylcholine, once it gets into the synaptic cleft, is going to migrate and diffuse across the cleft from a high to a low concentration. And it's going to bind to these receptors on the motor end plate. Okay, now remember, this muscle cell is normally sitting at -70 millivolts. More sodium out, more potassium in, negative charge inside the cell, positive charge outside the cell. Once acetylcholine binds to those acetylcholine receptors, we're going to depolarize our membrane. And how we do that is sodium channels open and sodium diffuses in. Remember, sodium has a positive charge. So that's going to make the inside of that membrane become more positive. We call that depolarization, right? Now, initially, this is just going to happen at the motor end plate. If we depolarize enough to hit threshold, remember the number for threshold is -50 to 55. If we hit threshold, we get a ton of sodium channels opening. And that's going to cause the inside to become very positive. it gets up to positive 30, and that's how we get an action potential, which is going to self-generate. It's going to spread down the sarcolemma and down the T-tubules. Once it hits those T-tubules, it's going to cause calcium that's stored in those terminal cisternae to get released. Remember calcium? Oops, I forgot to put a 2 here. Calcium is like the chastity belt, right? It unlocks troponin and tropomyosin from the active site. It allows the active site to be open, and now myosin can form a cross bridge. And it can push the actin filaments towards the midline. And that sarcomere can get shorter. And that's our contraction. And if this happens, the entire muscle cell contracts and gets shorter. Now, again, the only thing we didn't cover here is ATP. So remember, ATP is used in this process of getting that sarcomere to shorten up. Now, the very last steps 9 or 10, 11, and 12, this is undoing everything. So we've walked our way down to a contraction. So now let's undo it. So one thing we have to do is, if we kind of start down at the bottom, we have to get rid of calcium. Calcium's in the way. So I'm going to do this in red. So how we get rid of calcium? Terminal cisternae, just suck it back up like a vacuum cleaner. You get rid of calcium, and now the chastity belt goes back over those active sites. Myosin can no longer latch on, and the sarcomere lengthens back out. We have to undo this positive charge on the inside, right? Remember, we depolarized our membrane, we got an action potential. So now we need to repolarize our membrane. How do we do that? Close all these sodium channels. Think about what channels you got to open. Potassium. That's repolarization. So we're going to open potassium channels. Potassium has that positive charge, so the outside now is going to become positive, and the inside now becomes negative. And then the last thing we have to do is we've got to get rid of all of that acetylcholine. And we get rid of acetylcholine through that enzyme called acetylcholine esterase. And that will degrade acetylcholine. So those 3 steps at the end just undo everything that we did. So this will get us back to everything resting, no contraction, get us back to resting membrane potential, no acetylcholine in the synaptic cleft, and now we can do this all over again, okay? So if you can walk your way through and talk your way through all the way from an impulse coming down a nerve to getting the sarcomere to get shorter, and then walk yourself backwards to undo everything, like I said, you're good. You can answer all the questions that I asked you on muscle phys. Okay, so let's see. So this is a good slide to study, obviously, as you're doing your drawing, if you've followed along. Now, obviously, as I made that drawing, there were other things that I was talking about that didn't include the drawing. So when I say if you can draw it out, I also mean that if you can explain it, if you can draw it out and explain what's happening, you're good. Okay. So this next part that we'll do next week is on how whole muscles in the body contract. So not just individual cells, but like, how do you get your bicep to contract? What's happening there? How's your quadricep contracting? So this last part is looking at more like whole muscle contractions and muscle metabolism, okay? Does anybody have any questions from this part of chapter 9? Okay, draw. If you're drawing and you're not sure about something, you've got questions, let me know. I will see you all next week.